This evening, we're going to be hearing about results from on-farm trials that have been looking at different nutrient management practices and cover cropping on organic farms across southwestern BC. You're also going to get a chance to find out about a new four-year project that's going to be building on this research and offers farmers a new chance to participate in the research. I'm Shauna McKinnon. I'm the network manager for the BC Agricultural Climate Action Research Network. I'm joining this evening from my home on the traditional, ancestral, and unceded territories of the Coast Salish peoples, the Squamish, Tsleil-Waututh, and Musqueam nations, and I live in Vancouver. We're in meeting mode, so you guys can actually see who else has joined you. I'd love if people can just take a minute to introduce themselves in the chat. I won't make you put your videos on if you don't want to, but we also would love to see your faces, so don't feel shy. It's kind of like a casual fireside chat this evening, organic farm fireside chat. So yeah, feel free to have your video on or off. We are recording the session, but it's just going to be recording the presenters. Don't worry, even if you have your video on, we're not going to be recording you. And we're not going to be sharing the recording publicly this time. We're just going to share it with a few people who registered who weren't able to come tonight. Also, we're going to have a few breaks with Q&A, and we have the Q&A box that we can use for questions. And we will have time for questions and some discussion. I think from here, I'll just introduce our speakers this evening. So the research that is going to be shared tonight is coming from the Sustainable Agricultural Landscapes Lab at the University of British Columbia, Vancouver campus. You probably have already met these researchers, this team, because they have been working on these topics for quite a while. So Lisa Lewis, who is a co-owner and operator of a diverse 40-acre farm in the Cowichan Valley and is a part-time teaching and research faculty member at UBC's Faculty of Land and Food Systems, is here this evening. And she brings close to 30 years experience as a certified vegetables organic farmer and 17 years experience with soil health focus on farm research and field trials. So thank you, Delisa. It's great to have you here this evening. We also have Sean Smuckler, who is an associate professor and chair of agriculture and environment and director of the Center for Sustainable Food Systems in the Faculty of Land and Food Systems at UBC. He leads the Sustainable Agricultural Landscapes Lab. He's also the chair of the ACAR network, and his research focuses on developing ways to help farmers adapt to climate change and improve the sustainability of their farming practices, specifically as it relates to soil. And that's why he's got some soil in the background of his photo. We also have Relani Kessler, and she is a research assistant with the Sustainable Agricultural Landscapes Lab and an instructor in UBC's Faculty of Land and Food Systems. And Relani is going to be presenting on a few different research trials tonight, including research that she completed for her master's degree in soil science. And she also has a background working on organic vegetable farms. So welcome, everyone. It's great to have you all here. This is a repeat of a session, or very similar to a session that we delivered last Thursday morning. So if you were here for that morning session and things start to feel very familiar, we won't take it personally if you decide to jump off, because we are sharing the same results, although the questions will be different. Okay, so Lisa, Sean, Relani, great to have you. Sean, I think we're going to start with you, and I can pop up the welcome poll if that's where you want to start.
So we'll be talking about work that we've been doing over the last 9 years. But before I jump in, I do want to recognize that I am also joining from the unceded territories of the Musqueam, Tsleil-Waututh, and Squamish people, and recognize that my collaborators are calling in from other locations on unceded territories, and that much of the work that we've done across the province is also on unceded territories, and that we are incredibly grateful for having the opportunity to work on those lands. And we recognize that while our work is largely focused on colonial agricultural systems, that there are alternatives to these systems that we hope to support and provide recognition for. We also want to recognize that there are a number of key collaborators that were essential to this research, and recognize that this research would not have been possible without the funding from organizations like MyTax or the Organic Science Cluster, particularly the BC Ministry of Agriculture and the former Climate Action Initiative, as well as NSERC. And of course, there are a number of farmers and others that were essential to this work, including numerous students. And I saw Rachel Rusin has joined the call. She was also instrumental to this work. So Rolani and Delisa and I are going to tag team this. I'm going to start by giving a quick background on what regenerative agriculture is and its linkage to soil health, and then its linkage to the climate crisis. And then Rolani's going to provide some highlights from the trials that we've done over the last 9 years, and then Delisa is going to jump into what's next, talk about our new exciting project. So we already did the icebreaker. So let's talk about regenerative agriculture. So regenerative agriculture is defined in many ways and used quite often these days. I particularly like this definition: an approach to farming that uses soil conservation as the entry point to regenerate and to contribute to multiple ecosystem services. And I like this one because it centers specifically on the soil, and that it recognizes that soil is responsible for a number of outcomes that we as humans are interested in. And these are what we deem ecosystem services, ecological functions that are useful to humans. There are a number of ways to define regenerative agriculture. Some people define it based on principles. These are typically the principles that you see used to define regenerative. So maintaining a living root system, minimizing soil disturbance, maximizing crop diversity, keeping the soil covered, and integrating livestock. At the same time, there are a number of practices that are often pointed to as the definition of regenerative. And those practices can include conservation tillage, cover cropping, complex crop rotations, residue mulching, integrating either nutrient management or pest management into the system, or adding organic amendments, or a combination of the above. I think it's really important to recognize that what we are really looking for are outcomes. And one of the key outcomes from regenerative agriculture that we're looking for is improved soil health. And when we talk about soil health, it's a framework for us to evaluate the capacity of the soil to provide those different ecosystem services that I was just talking about, ecosystem services that are vital to plants, animals, and humans. The main process that we're focused on when we're either defining or the practices or the outcomes related to regenerative agriculture are related to the way we're incorporating carbon into the soil. So through photosynthesis, plants take up carbon dioxide, translocate that carbon down into the root system, and feed the soil biota. The exudates from the plant roots that provide the basic sustenance for those soil organisms eventually gets transformed into soil organic matter. And soil organic matter is sort of the linchpin for what we often are using as a set of indicators for soil health. And 50% of that is made up of that carbon that was taken up by those plants. So when we're thinking about healthy soils have their own set of outcomes that we're looking for. Outcomes such as regulating water or sustaining plant and animal life, filtering pollutants, cycling nutrients, or just basically providing physical stability and support for the plants that we're trying to grow there. There are, however, a number of challenges when trying to improve soil health that need to be recognized. And these are particularly challenging in mixed vegetable farming. Some of these challenges include the emissions of greenhouse gases. When we're providing nutrients, particularly nitrogen, to the agroecosystem, it's an inevitable outcome to emit nitrous oxide, a powerful greenhouse gas, to the atmosphere, or when we're disturbing the soil, we're releasing carbon dioxide. If we're adding nutrients to the system, those nutrients may be susceptible to leaching, or if we're adding phosphorus to the system, we're often building up phosphorus over time. We have constraints of labor, we have constraints of access to nutrients, cost of transporting of those nutrients. And in areas like the Lower Mainland in particular, we have limited land base that constrains our operations. So when we're thinking about the relationship between, in particular, some of these outcomes and the climate crisis, we're thinking about trying to improve soil health so that we can actually mitigate some of the negative aspects of these practices on the climate. So ultimately, we want to be able to sequester carbon dioxide out of the atmosphere and store it in the soil. But we know that any disturbance of the soil also releases carbon dioxide back into the atmosphere. So what we're after is a net positive carbon sequestration and an increase in soil organic matter. We also have to recognize that there are these other greenhouse gases, methane, and as I said before, nitrous oxide, which is almost 300 times as powerful at warming the climate as carbon dioxide, that are also being emitted from the system. So we have to think about this in terms of a net greenhouse gas emission and impact on the climate. So thinking about soil organic matter in the context of these other outcomes. So we started our research at the UBC farm back in 2015 with a project that was led by Gabriel Matthias Laundry, a postdoctoral fellow at the time. And he did some basic research on a number of different soil amendment practices for organic vegetable production. And while the amendment treatments weren't very unusual, what Gabriel showed us is by measuring a number of different outcomes at the same time, he highlighted that there were trade-offs that were very different depending on what kind of approach the farmer was using. So this radar graph, as we call this, illustrates that in some cases, using lots of compost had positive outcomes in some indicators, but not in others, whereas using manure had benefits in other cases and not in the same ones. So following that, Kira Borden and Amy Norgard led a study that again was on UBC farm, looking at a similar set of nutrient management approaches. And then we started another experiment very similar to that one at Greenfire Farm, where Delisa has her operation, and then replicated a subset of those treatments on 19 farms across the province. Following that, we built on that experiment, that set of trials, and looked at overwinter covers. This was led by Rolani Kessler and Carson Lee at the time, and looked at a similar set of treatments and a different set of farms in different locations across the province. And what we're going to talk about tonight at the end here is the next phase of this, looking at tillage as part of this management approach and different covers, hoping that we can help address some of the trade-offs that we've seen to identify synergies and optimize for overall soil health benefits. And the goal would be to continue to use Greenfire and UBC Farm as mother farm experimental sites, and then to establish trials on 15 additional farms across the province.
And Raylani, you could get started. Does that sound good?
Yeah, sure thing. Thanks, Sean, for the introduction to all the studies and also to the context where we're working. And we're going to transition now into some of the results that we've found from some of these projects we've been working on for the last few years. And all of the different projects have slightly different management practices that are the areas of focus. And some of the ones we'll be looking at today are soil fertility, so optimizing compost and fertilizer applications, and then also different avenues for soil cover, which include cover cropping and tarping. And there are components of each of these studies that will be carried forward into the part that Delisa will talk about in the upcoming trials that are starting this year. So the Organic Vegetable Nutrient Management Project, which ran 2018 to 2019, was aimed at evaluating nutrient management strategies to improve nutrient balance and yield in annual vegetable crops. And this was done by focusing on two general fertilizer or fertility strategies. One was a high application of compost, and the other one was a small application of compost with an additional application of nitrogen fertilizer. And in this trial, we used a mother-daughter field trial approach. And on the two mother farms, we conducted more intensive experiments with on-farm replication and more frequent sampling. And the final results that I'll share, or the first set of results that I'll share here, come from these mother farms, which are UBC Farm in Vancouver and Greenfire Farm in Duncan. And we'll look at some data from the regional farms in a minute as well. So the amendment strategy that we use, as I mentioned, was this high compost versus a low compost plus fertilizer. And these were two different methodologies, both aimed at getting the plants what they need. So the calculation here starts with what we expect a crop will remove over the course of the growing season. So like what's the crop requirement and then amending to that level. And when you amend with compost, there's a higher proportion of phosphorus than nitrogen in compost. So when we go to meet that nitrogen demand, we end up over applying phosphorus. So in this high compost strategy, we applied enough compost for the nitrogen demand to be met, knowing that there would be more than enough phosphorus to go around. And in the low compost strategy, we applied compost just to meet the phosphorus requirement of the crop and then added an organic fertilizer to meet the nitrogen demand. And this was feather meal in this trial. And then we also compared both of these strategies to a control. So just a baseline with no fertilizer or compost amendments at all. And when we compared these two strategies relative to the control in terms of yield, we found that the high compost application produced about 50% more yield than the control plots, and the low compost plus fertilizer had about 40% more yield. However, we also found that the yields in the high compost plots were highly variable, and the low compost plots were far more consistent. We also measured nutrient use efficiency, which tells us how much of what we applied was actually used by the crops. And here we found that in this high compost strategy, about 4 to 15% of the nitrogen from the compost was recovered by the crops, whereas 30 to 53% of nitrogen was recovered by the crops in the low compost and fertilizer treatment. For phosphorus, we found that less than 5% of the phosphorus was recovered by crops in the high compost and 20 to 64% of phosphorus was recovered in the fertilizer treatment. So these are all statistically significant differences in the ability of the crops to pull up what we were putting down based on amendment strategy. This experiment was also conducted in a modified way in 3 regions. So there were farms on Vancouver Island, in the Lower Mainland, and also in Pemberton Valley. And these are what we call the daughter farms. So each farm only had 1 replicate of the treatment per farm, and the type of compost and fertilizer varied by farm. We generally used whatever they were using. And on the daughter farms, we tracked input costs for the compost and fertilizer. Here we're looking at a graph of the input costs. And so we have on the y-axis, this is the cost in kilograms per hectare. It must be dollars. And then we also have the different regions on the x-axis here. So the Fraser Valley, the Pemberton Valley, and Vancouver Island. And the colors here represent the different strategies. So this red box plot is the high compost, and then we have the low compost plus fertilizer is blue, and then the typical approach of that farm is in yellow. And the black dots here are the means, so that's what we're using to compare. And the main takeaway for us here is that the input costs were highly variable across the regions, and this was primarily driven by the cost of compost, which was significantly more expensive, particularly on Vancouver Island, than in some of the other regions. And that trend holds here. So if we look at Fraser Valley, Pemberton Valley, and Vancouver Island, the high compost treatments are all quite different. And there's also a bit of a difference in how the strategies we were trialing compared with the cost of the strategies the farmers were already using on the farms. In some cases, they were less expensive, and in some cases more. From this trial, we took away that we were achieving more consistent and high yields using the low compost plus organic fertilizer methodology. The high compost had a similar boost to yields, but there was more variability across the plots. We also saw that the low compost plus fertilizer had higher nutrient use efficiencies. So the crops were using more of the nutrients from the fertility applications, but we don't know about the long-term impacts on soil organic matter using this strategy because we are applying less compost, so less organic matter overall. We found that this approach of trying to do the balance, starting with what we think the crop needs and then backtracking from there to calculate what our applications should be, was promising in the system. And we have since modified the approach a bit. We built a calculator so that we can also account for the nitrogen that's already in the soil, and then the nitrogen that we expect to be contributed from an overwinter cover crop, so we can better make the calculations of what to apply prior to the growing season. And then lastly, we saw that the inputs were highly variable across the regions in this trial, primarily driven by compost costs. So the next trial we'll talk about here is the winter soil management trial, and this ran 2020 to 2021. And here we were evaluating winter soil cover strategies to determine the impacts on spring soil water conditions and also spring soil nitrogen. And we focused on 2 strategies. So we were looking at cover cropping and tarping with silage tarps, which you can see depicted in this photo here. And then we also ended up with a comparison of the tarps to kind of a winter fallow situation or a sparse cover crop. And this trial had a similar design to the organic vegetable nutrient management trial. We had 3 regions where we had daughter farms. The mother farms were the same, so it was Greenfire in UBC. And then we had daughter farms in Vancouver Island, the Lower Fraser Valley, and the Kootenay Mountain region. And one of the big takeaways for us here was in spring soil nitrate. So let's walk through some pieces of this graph here. We have nitrate, which is a plant available form of nitrogen that we find in the soil. And this is measured in kilograms per hectare. Here we have our results from 2020 in the top graphs, and there's results from 2021 in the bottom graphs. UBC and Greenfire each have their own graphs here. You can see along the x-axis, and then the Kootenai Mountain, Lower Fraser Valley, and Vancouver Island farms are all grouped together on the far right. All of the black triangles are the soil nitrate levels that we saw underneath the tarps. And then the green, either circles or squares, were a cover cropped plot or something that we attempted to grow a cover crop in that didn't take off for one reason or another, either early winter or waterfowl or whatever reason. And across the board, this measurement took place in the spring. So we took these tarps off in mid-April after they had been down since either October or November. And then we took a soil sample and measured the nitrogen then. And in every case, we saw that soil nitrate was higher in the areas that had been tarped rather than the cover cropped or the untarped areas. And this was a significant amount of nitrogen agronomically. We grew beets one of these years. So I'm going to use this crop as an example, but in the higher range of our measurements here, the nitrate that was underneath the tarps represents more than 70% of what would be recommended to apply for a beet crop in BC. It's important to keep in mind that the residual nitrogen in the spring would be related to whatever got applied in the following year. So this really depends on the fertility application of the farm. And also we took these measurements before the cover crops had been turned in. So we would expect some nitrogen to come from the cover crops as well. This is a graph from UBC Farm where we measured soil water.
This graph is highlighting the water volumetric water content over the course of the 2 years. And the shaded areas are areas where we had we were running our experimental covers. So we used the silage tarp and cover crops. And when you zoom into those areas in the red, which highlights sort of a flex point before the production season starts in the unshaded area, you can see that there's a key difference in the water under the tarps. So during the winter, as one would expect, the tarps are shedding water, preventing water from entering the system and actually keep the soil dry. And then there's a flex point where when the tarps come off, the tarps are actually the cover crops then switch and then are drying out the system much quicker than the tarp system. The cover crops are basically pumping water out through photosynthesis. And then that flex point, that difference lasts for a few weeks into the production season. So what this illustrates is clearly the tarps play a role in water dynamics, but it also highlights that the water dynamic, that outcome that a farmer might want, really depends on perhaps the timing of the precipitation, the subsequent crop that's going to be planted, how long they want the soil moisture conditions to continue. And one thing that should be noted is that the UBC farm is a very coarse textured system. So while these conditions might persist for 4 to 5 weeks, on our farm, it might be very different on different soil textures. How are you feeling, Rolani?
Yeah, thank you. Good thing you've seen that graph like 100 times before. Yeah, and the only thing that I would also point out is that on this farm, that inflection point happened in like the first week of March. on both years, but that would be like really variable depending on the soil type and the precipitation on the farm. So from this study, we took away some key points related to both the nitrogen and the water. The first was that the tarps that we laid down were effective at preserving nitrate over the winter, and that the timing of the tarp removal was really important for soil water conditions. So as Sean said, the tarps helped keep the soil dry over the winter, and then at a certain point, they were really working to keep water in, where the uncovered areas started drying out much faster in the spring. And we also had left the study with a lot of questions about the long-term impacts of these practices. So again, one about soil organic carbon. Cover crops are adding carbon to the soil, whereas these tarps are not. So what that would look like over the long term is kind of uncertain. We measured some yield metrics in this study and we didn't see a big difference, but we also didn't take into account ideas about labor or weed suppression or the other ways that these tarps would be used on farms that might impact yield. And we didn't use them as a reduced tillage measure, which is something that could also impact organic carbon, and a way that a lot of farms are already using these tarps to limit or cut out tillage in the spring entirely. And the last study we have to share today is the summer cover crop trial, which we conducted last summer. And this is something we did as kind of a pilot study for the project that we're launching this year, when we knew we wanted to have a summer cover crop treatment in our upcoming experiment. And we wanted to know which species or combination of species might be a good fit on the farms that we were working with. So we evaluated cover crop mixes for their contribution to soil nitrogen, weed suppression, and their performance without irrigation. And we selected 3 species. So we have oats, peas, and fava beans. And we tried them in 6 different mixes. So we have a mix of all 3, oats, peas, and favas, and then the combinations of 2. So oats and peas, oats and favas, and peas and favas. And then just peas and just favas. And these were selected for their regional suitability. And we also wanted to try, one of the main goals of this is to have these summer crops, cover crops be contribute to the nitrogen pool in the soil. And so we knew we wanted to have at least 1 legume, maybe 2. And then the oats we were hoping would be a big contributor to biomass and also weed suppression. So we trialed these different treatments on 3 farms last summer: UBC Farm, Greenfire Farm, and then also Amara Farm in Courtenay on Vancouver Island. And all of the cover crops were planted between May 25th and 30th. And then we collected the biomass at the end of July or early August, which would be like the termination time for these cover crops. So these are figures of the total biomass put on by the different cover crops on each farm by irrigation. So the blue dots were the irrigated plots, and the gray rectangles or triangles are the unirrigated plots. So pretty unsurprisingly, when we watered these plants, they grew more. It was about double the amount of biomass that they put on. However, in the unirrigated trial, there was still a pretty significant amount of biomass. So about 17,000 pounds per acre on average across all the treatments. We did see some differences by treatment type. So notably, the favas by themselves put on the least amount of biomass. And then all of the combinations of 2 had fairly similar biomasses. When I looked at this, like by treatment, but not by farm, just like how the treatments performed across all the farms, it seemed like peas were a large contributor to the biomass contribution itself. They did really well. At Amara Farm, some of you might have been in the same position last summer, but there was not a lot of water available, and they ended up not being able to water the irrigated part for the whole trial, which is why these you don't see much of a difference on that farm. Another thing we noted is that we didn't see a meaningful difference in maturation time of the crops between the irrigated and unirrigated trial, which is important to us because when we estimate the nitrogen contribution of a cover crop, it's really dependent on what the phase of life the plant is in. So if things were maturing really quickly under unirrigated conditions, we would want to know that so we can best time when to terminate them for maximum nitrogen contribution. These are figures of the weed biomass from UBC and Greenfire. And here we also saw that the irrigated crops, for the most parts, grew more weeds than the unirrigated crops. Notably, the oats were, in fact, important for weed suppression as we had thoughts. However, we also noted that having a 3-species mix, apart from like a 2-species mix, didn't make a big difference in terms of either weed suppression or total overall biomass. The seeding rate was the same for both of these irrigated and unirrigated trials, and that is also one thing that might contribute to a bigger difference in terms of weeds or biomass, because we would expect less to germinate in an unirrigated trial versus a irrigated trial. So if you are going to grow unirrigated cover crops, you may consider increasing the seeding rate to get some of these same advantages of an irrigated trial. And one last thing to note here, because favas get like a really bad rap in this graph series in particular, definitely the seeding rate that we used was on the more conservative side. And if we were to do this again, I would probably increase the fava seeding rate by maybe 3 times. And then I think it would have held up a little bit better against some of these others. So the takeaways from this trial was that even when unirrigated, the summer cover crops put on a significant amount of biomass in the 3 months that we had them growing. The peas and the oat pea mix were the most promising for a short season cover. They had the most biomass overall for the greatest weed suppression bang for your buck, and also were the least expensive seeds. And as I mentioned, we would increase that fava seeding rate if we were to do it again. We have a bunch of samples of these cover crops that we're testing to determine the nitrogen content, and then we'll be able to do calculations to determine how much nitrogen we would expect to go into the soil that could be used by subsequent crops. But just by looking at these cover crops when we went to terminate them, they were all further along than we would want them to be for maximum nitrogen contribution. So it would be helpful to terminate them earlier if that was a primary goal. And we also have an upcoming comparison in this trial to a 1-year cover. So these are crops that we planted last May that are perennial grasses and legumes that we left in the ground for 1 whole year. And we're going to compare the biomass and weed suppression and nitrogen contribution of this strategy as well. No, that was a lot of information. Do we have time to stop here for questions, or should I just answer them in the Q&A and let Delisa take it from here?
I think we have time for a few minutes for questions. Yeah. Okay. So I'm going to go back to the first project that you shared around the compost trials. And do you have any theories on the variable yield response that you got out of the high compost treatments?
Sean, do you want to take that one?
Yeah, I mean, I think that's an example of how variable compost can be. I think when we're moving compost from a large pile that isn't thoroughly mixed before going on the field. And I think this is an example of how that type of practice can lead to variability in the nutrients available. We really do our best once we're like that. The compost that we apply by hand to the field. And so we do a really, I think, good job of making sure that compost is equally applied across the plots, but we don't have any control really about how thoroughly the compost is mixed beforehand. And even if you are mixing compost really well, I imagine there's still quite a bit of variability in that. Delisa and Rilani, something to add to that?
I think the variability is the key, and it's also that the plots themselves vary in their field position. So this replicated design, as well as the variability of the compost, would be sort of my top choices for that variability reasoning.
Yeah.
Yeah,
Great.
and I think the only thing I would add to that is that when we're making these calculations, we are making an assumption about what percentage of the nitrogen that's in that compost that's actually going to mineralize in 1 year. So whereas with the fertilizer, it's less of a guess in terms of how much of that nitrogen actually makes its way out and is available to the plants.
Great, thank you. I do want to take 2 more questions before we move on, if you guys don't mind. So the first is just around whether or not you included soil microbes at all in your analysis. And yeah, if you could just speak to that, especially like this question came up around the cover crop trials where you were looking at nitrogen fixing crops as well as non-nitrogen fixing.
Yeah, go for it.
Yeah, I was just gonna say in those trials, we didn't do any direct microbial analysis. We did at the UBC farm measure carbon dioxide fluxes from all of our plots. We didn't show that data here, but there's clearly differences in carbon dioxide, which is a measure of microbial activity. And since these studies, we've integrated a soil indicator that's based on the flux of carbon dioxide for each of the samples, regardless of whether we're measuring them in the field. So it's an in situ laboratory analysis. But no, we don't. Other than that, we're not doing microbial analysis.
Okay, great. And oh, sorry, go ahead, Rilani.
Oh, good. There was a trial that was related to that tarping and cover cropping trial that I talked about that happened at UBC Farm, where they did like mesofauna and macrofauna analysis that was looking at the differences between the tillage and types of soil cover. But I don't think we have that data yet.
No, not yet.
Interesting. Okay, I think our discussion wouldn't be complete without mention of wireworms, right? If we're talking about organic vegetable production. So the question is if wireworm or other pests was a consideration in choosing legumes versus oats. And this is a question coming from the Cowichan.
Oh, wow. It hurts to have a question about wireworms that isn't for Delisa. I feel like she's always the one advocating for the thought of wireworms in these treatments. In the cover crop piece in particular, that wasn't something that we thought about or like used as our criteria for entry or for selecting. But maybe Delisa, do you want to talk about the other ways that we think about wireworms?
I mean, I'm pretty sure there was a hard request for not fully relying on rye as one of the
That's true. Yeah.
choices here for that very reason, because of the wireworm buildup we've seen here in the Cowichan Valley when rye goes too woody. So it was considered in that sense.
Great. And so I think for the rest of the questions we have in the Q&A, I'm going to get you guys to answer them by chat or by type. So don't worry, we're going to get your questions and you can always stay on if you have lots of questions. I think our speakers would be willing to stay on a few extra minutes. So I'm just sharing the results from the poll just now. Delisa, while you get set up there. So also your poll results will be shared with this team and they are considering your answers for the research. So I think everyone should be able to see those if you want to just take a quick flip through and see what other people rated as the different priorities, what kind of practices they're using. And Delisa, unless you want to refer to these, I can stop sharing it now before you start.
Well, thanks so much, Shauna, for that. And to everyone for showing up and sharing again, particularly those of you who've come back again for a second visit with us during this webinar. We really appreciate all of you. It's great to see the numbers of farmers on a percentage basis who are here, because the next part of this is going to be introducing basically the follow-up, the regional on-farm field trials that will be part of the Organic Science Cluster 4. Sean introduced you all, and Rilani as well, to some of the research results from the Organic Science Cluster 3 research, the organic vegetable nutrient management study. And so what I'm showing you here are some of the questions we hope to address that have been built upon these 9 years of field research looking at soil health. And that is one of the reasons we're bringing in this question around the reduced tillage. But much of what we'll be doing, great to see your responses to the polls around some interest in reduced tillage, as well as the targeted nutrient management and cover crops. So particularly for these regional on-farm trials, we'll be including the regions of Vancouver Island, the Lower Mainland, and the Okanagan. And again, great to see in those polls that we've got representation, some people here coming in from those spots. Let me just move on to, I want to talk a little bit about the research design. And again, this is building, very much building on the work that has been done these last years. So we'll be looking at 5 regional farms in these 3 regions that I just mentioned, and looking to evaluate strategies for reducing tillage again on these organic vegetable farms. Our aim here is to recruit and share information and encourage people who are interested in participating with us in learning more about this. The overall aim is to help organic farmers around reduced tillage. Or we know that organic vegetable farmers in particular face something of a dilemma with efforts to promote soil health, particularly soil health outcomes, relying on mechanical disturbance, conventional or turnover tillage as one of the mainstay components for crop establishment, as well as weed control. I know I do. So coming very clean about my own tillage practices. And similarly, the approach to nutrient management will build upon those studies that Rilani shared results with you on. We'll be looking at comparing a typical approach, how you manage soil health nutrients on your farm, and looking at that compared to that sort of balanced in, balanced out, what goes in and out, targeted approach to nutrient management. And again, leaning into some of what we've been learning around ways to grow nitrogen on farms with the summer cover crop and compost studies as well. All of this kind of a suite of management practices that are aimed to help us minimize those trade-offs and optimize outcomes on organic farms for soil health. I want to just note a little bit the roles of the collaborating farmers, but we do have a one-pager description of the upcoming trials. And when you reach out to us with interest, we'll be sending and sharing that with you. We'll also be sending and sharing it with our sort of partnering organizations. So again, one of the key opportunities or farmer benefits that we want to create and facilitate with this 4-year and upcoming project is the opportunity to learn about reduced tillage and nutrient cover crop management from and with other organic farmers around the province. That would be a sort of shared and knowledge transfer plan there. So What the farmers would contribute, of course, is a small area of field, and we would ideally be looking at 4 years, all 4 years of the project. And those farmers would be helping to set up the trial and coordinating a little bit around what we found to really facilitate the research, that is a single harvest crop. And the example that you've heard already tonight, Relani gave it, the beets. That's one that's upcoming in our rotations right as this season begins. So beyond that, I want to look at just a quick note on the research principles guided by agroecology here. We're really looking at this contextualized, and in this case, regionally contextualized research. It's meant to be targeted. It's also meant to be co-created and facilitate opportunities for co-learning, embracing that complexity that we see in these on-farm research trial designs, and learning from that and being adaptive. That's the purpose here, is being a part of learning into adaptations to the climate stressors that we're facing on farms around the province. So rather than going too deep into this participatory research, note that, again, what we're doing here tonight is an invitation to you. We're at a choice point. We're hoping to partner with farmers around the province and ultimately end at a place of, collaboration and co-learning with this research. And so this question, many of you have already answered in your polls. I'll pause for a moment so we can have a few minutes that are still within the hour for any questions from the audience.
Thank you for tonight's presentation. I'm interested because I think we have some certified organic farms who would be interested to be a sister site. Certainly they face the same challenges you're all seeing, and we could probably partner through one of the regional extension committees, because we have the same problems, except, knock on wood, no wireworm.
Yeah, Nicole, it'd be great to get a list of folks that you think might be interested in participating, or just share our information with them directly.
Great, I'll do that. Thank you.
So there is a question that just popped up in the Q&A, and it's about the incorporation of livestock in the cover crop trials. And there were a few people that mentioned in the chat that they do have mixed production. Who wants to take that one?
What is the question?
I think it's it's a great question. It's whether we'll be incorporating livestock integration in the trials. And I think the answer is not this year. It's certainly a dream of ours. And if you're interested in our doing that, please let us know.
Yeah, one of the key objectives of this this set of experiments that we're about to launch is to try to figure out how to cycle nutrients on site as much as possible. And livestock certainly seems like a great approach to doing that. We at UBC Farm are in a transition right now, and we don't have the livestock to do that experiment.
There aren't other questions popping up now. You have Relani's contact information. She can connect you with the full team. And I want to say a huge thank you to our presenters, also to everyone that took time out of their evening to join us tonight. So great to see people joining from across the province. And yeah, thanks everyone. And I look forward to seeing the results of this new research and seeing the expansion of these trials into new parts of the province, and just seeing you build on the practices that you're studying.
Thanks so much, Shauna. Thanks for hosting us.